Good morning. We are at TAGC 2024, and this morning we are going to extract DNA, talk about DNA, and highlight a paper that was a joint project across 24 people, including my lab, that was driven by an undergrad initially in the lab, and we're going to talk about how you make science communication more accessible. We have two students from Laurentian with me. Jesse, do you want to introduce yourself?
Hey everyone, I'm Jesse Petahtegoose. I am a student at Laurentian University. Been there since about 2017. Joined Dr. Merritt's lab 2018. I'm now a master's student in his lab. And this is Allie.
Sorry. I'm Allie. I'm from Louisiana, Ontario, and I have been at Laurentian University since 2018 and have been in Dr. Merritt's lab for the past 3 years.
Thomas is fine. Okay, so we're going to go through, we're actually going to do a DNA extraction. This is published in a journal called Core Source, and the DNA extraction that we're going to do this morning is pretty straightforward. And it's one that many of you will have done before. I did it with my kids' preschool class, and I've done it at nerd nights in a bar. So there are lots of different ways that you can approach the DNA extraction. We're going to do it this morning using bananas. And one of the first things that we always talk about with this is it really doesn't matter what you use to do the extraction. So we have done this with saliva. When we do it in bars, we spit into shot glasses and you extract your own DNA. And you can do it with bananas. The amazing thing about life on this planet is we all share the same information. And that molecule that holds the information is DNA, and that's what we're going to extract this morning. So everybody take a bag. I'm going to divvy up some banana, and we're going to get started. So you two were both part of the project. Were you part of the project early on when we first started? I don't remember when you came in. So what did I pitch to you? What did I say to you we were doing?
That we were making a teaching tool to teach people about DNA, and that's essentially the gist that I got out of it.
Okay. Yeah. And
I already have a question.
yeah.
Remember your talk about transvection 2 days ago? It's on Twitch. What motivated you to go back to something as, quote, basic as DNA extraction, publishing a paper and teaching people?
Yeah, all right, that's awesome. So we get a lot of money from the federal government to do the science that we do. And one of the things that I take very seriously is that we have an obligation to explain to the people that are paying for this research what we do and why we do it. And if I go into a public space and talk about capitalism and fruit flies, I don't engage 5% of the people. If I go into a space and I say, hey, we're going to start with a piece of fruit, with saliva, with blueberries, strawberries, bananas, and we're actually, by the end of this, we're going to hold at the end of a stick DNA. And in some cases, we're going to hold our own DNA, right? I mean, and that's one of the really neat hooks. I just didn't want us all spitting in shot glasses at
And just like looking at everyone at home going,
yeah. So, okay, we're at the first step. So the short answer is because DNA extraction is a really engaging way to talk to people about science, and talking to people about science matters because everybody should understand what we're doing and the value of what we're doing. Okay, so what are we doing right now, Alan?
Adding water to it?
Okay, what'd you do before? What'd you do before the water?
Oh, crushed it.
Okay, why?
To break up the big pieces and turn them into small pieces.
Yeah. And so what we're going to do over the next 10 or 15 minutes as we do this, the DNA is inside the cells. So cells make up organisms. Complex organisms have lots of different kinds of cells. Bananas actually are relatively complex. But in all cases, the DNA is inside the cell. And so what we're going to do is a series of steps that are going to take us from our happy banana to DNA at the end of the stick.
so you take a banana or whatever you want to extract DNA out of, put it into a Ziploc bag, mash whatever that is into a until you're satisfied that you're going to be able to go on to the next step. Science is sometimes a little more art than science.
So all of these steps, so when we mentioned writing the paper, what we did with this paper was sort of put together a flow chart. I can hold it up here. And that flow chart allows you to walk through all the steps. And what we actually did was set this up as a choose your own adventure. And so in each of the steps, you can choose to do something slightly differently. And at the end of it, compare, did you get more DNA, did you get less DNA? And we'll talk at the end, it's not all DNA, but there's a lot of DNA in what we get at the end of this. So we have these handouts that we can be referring to, and we're using that to help guide us through. As a teacher, you can use this as a way to, in the classroom, work with the students. There are students that you're going to talk to, and they're going to understand what they're doing. There are other students that want to see that flowchart as well. And so the idea of having that flowchart is it's material you can give to the students ahead of time. It allows students that work really well off of handouts to work off of handouts, and then you talk to students. So we've taken our banana, we've mashed the banana, and we've now added, so immediately we're up to variations. I've got a lot of water, Jesse's maybe somewhere in the middle, and Allie's somewhere in between. And we're going to see at the next step that that's going to impact the amount of material that we're actually extracting. So we've got the bag, we've got the banana, and we've got a lot of water. Let me see which.
Actually, folks are asking, can we use any kind of water? For example, tap water might have different minerals in it that could impact the result.
What do you think?
I think you can use any type of water.
You can absolutely use any type of water, but it's a really interesting question because when we do this for real in the lab, when we do this and we're really, say, quantifying the amount of DNA, one of the things that we will change is the ions that are in the water. So we'll use different buffers to get different things in the lab. So that's a really sharp question. This is a really brute force extraction, and any water is absolutely fine. In this, sorry, go ahead.
Could I use my coffee?
I wouldn't use your coffee. Although, if we tried to extract DNA from coffee beans, we could do it with green beans. We couldn't do it with the roasted beans because the DNA is all been incinerated. One of the things you will find when you go through and like, you know, what's around the house. So I've done this, actually, I did this years ago with my kid. And we literally like grabbed the fruit bowl, like let's extract DNA from everything in the fruit bowl. And different kinds of material have more or less DNA in the material and are more or less easy to squash. And so it ends up, oranges are not particularly easy to squash in a Ziploc, bananas, strawberries, that kind of thing. All right, so let's close the bag for a second and we're gonna make our...
Here's one follow-up question about the water. Sparkling water, yes or no?
Oh.
Ooh.
I would not. So, and you will see in a second why sparkling water, it shouldn't impact your results, but at the stage we're gonna get to, you really don't want fuzzy water. Okay, so we've got our sample and we have mashed it. You want to mash it, like I didn't really mash mine as much as you should have. You don't want any large pieces. So the DNA is inside the cells, so you want to get this to the point where when you hold it up, it's sort of this uniform slurry and doesn't have the big chunks that I have. We're using so much material that this is not precision science. Okay, so the DNA is inside the cell. We have mashed up the banana, and first, the next step is we want to remove, I think I want you there. We want to remove the solids because now the DNA is in the water. So one of the cool things about DNA, excuse me, is it exists in the water in our cells. And so when we break the cells, the DNA basically just starts to spread out in the water. At this stage, the DNA is still inside the cells. Okay. So we're going to make our filters.
You can just use a coffee filter.
Someone was asking what kind of cup filter for coffee.
A clear one would probably be better. for this extraction. For you can use a coffee cup.
So, like, I'll hold this up. What we've done is we've taken just a plastic cup, a basket coffee filter, and a rubber band, and we've rubber banded that filter across the top. And we're just making a physical filter to grab that. You can do this with anything you've got around the house. As Jesse was saying, if you can see inside, you can see things dripping, we'll hold that up. It ends up, one of the things that we found is that the cheaper the coffee filter, the better this works. So cheap coffee filters seem to drain really well, and so these are dollar store coffee filters. Okay, so we're going to, at this stage, actually, you know what, we can, I'm gonna look at our flow chart. We're gonna do a number of different steps, and you can do the steps at different times. We're about to filter. Before we do that, if we follow our flow chart, we're gonna add some salt. Yeah, and so what I was gonna say is, you two may not have enough water. If you want enough water, yours is,
That some of this liquid will drip through.
that's right. So it can't be too thick. If it's too thin, what happens?
Dilute it.
Yeah, yeah. And so you may have, you may not get as great yield at the end of it if it's too little.
We're scientists, we'll see how this goes.
Absolutely. And I'm struggling to open salt, which is not, there we go, okay. Okay, so DNA is this long molecule, like really long. The DNA in a human being is essentially 2 meters. So in translating that into non-metric, it's like almost between 6 and 7 feet.
That's in each cell.
In every cell. And in any organism. So, and the amount of DNA, the length of DNA varies across organisms, but you can sort of ballpark the idea that we're looking at 6 to 7 feet or 2 meters of DNA in every cell in an organism, and millions of cells. So in the banana chunks that we've got here, we had millions of cells of the banana, and we're trying to extract out of that this DNA. So it's incredibly long. It's so incredibly thin that you cannot resolve it. You can't see it. So what we're going to see is a mass of these incredibly thin threads, but they are in fact so thin that you can't see them with a light microscope, you can't see them with an electron microscope. The best that we can do is crystallize them, and at that stage, X-rays will actually be bent as they pass through the crystal. But it's mind-boggling just how thin and long the DNA molecule is.
So if you're following along at home, you've taken a banana, you've mashed it in a Ziploc bag, you've added some water, enough that you think that you'll be able to pass that water through a filter. Then that water is going to be holding your DNA. You're going to be adding a little bit of salt to that bag, mixing it up.
That's it. And
Question right away is, what kind of salt is this? Table salt? Is this?
Yep.
Can it be low sodium?
So, all right, and actually, so that was the next step. So, Allie, why are we using salt?
The salt is I think it is for the ion.
That's
so
right.
DNA is negatively charged,
Yeah.
and then the positive sodium will neutralize the backbone.
So table salt is the short answer. And the reason that that's the answer is that DNA, this long thread molecule, is negatively charged. And that negative charge means that individual DNA molecules tend to push each other away. And what we're trying to do is get them to clump. And so one of the steps that we're going to do is when we isolate we begin to isolate the DNA, we want those threads to clump. And so we're going to coat them with positive ions that are going to neutralize the negative charge of the DNA. And so that's the job of the table salt. So table salt dissolves, you get sodium ions, chloride ions, and the sodium ions are going to coat, which are positively charged, are going to coat that negatively charged DNA. All right, so we're now all pouring into our filters. And now we're at the this could go well stage, which means there's also it could explode. It's not going to explode, but every now and then the filters burst, especially if we do this with preschool kids. So we're going to let this drain for a minute, and then we're going to probably hurry it along by giving it a bit of a squeeze. So we've got this incredibly long, incredibly thin, positive or negatively charged molecule where we've got it in the cell. So at this stage, the DNA is still trapped in the cells. And in plants that you've got a cell membrane, you've also got a cell wall. The first step in breaking that down was to physically squash up the banana. We're now going to filter out the solids. Yeah, and so you can see at this stage, like the value of having a little bit more water in the extractions. If you hold these up, the amount of...
Well, yours is mostly water.
I don't know if that comes through on the camera.
Cells.
So, yeah. So on the one hand, you get a lot of water, things are really dilute. On the other hand, if you don't have enough water, it takes a long time to filter through. I did this with preschool kids.
Yeah.
And I was really, every now and then it doesn't work. Like, we'll see. The success rate is pretty high.
Oh, wow.
I want to say like 80 or 90%. But every now and then, like, okay, it doesn't work. So with these preschool kids. Oh, no, you're great, you're great. But I really didn't want it to fail with preschoolers. And so I was like, we're going to use like a quarter banana with every kid to make sure there's a ton. We ended up with Jell-O. And so we did the extraction, and the DNA did not precipitate because it was solid. It was like this viscous mass. And the kids are like, yay, slime! I'm like, that's right, that's science right there. And the teacher's looking at me like, is it supposed to do this? No, it is not. All right, so why don't we talk a little bit about the paper? So as we're waiting for these to drip, we'll give it a couple of minutes. The idea was to come up with a standard protocol that we could use to teach, and that a teacher could go into a classroom, a public speaker could go into any place and use this extraction as an icebreaker to talk about genetics, talk about DNA. The student who had taken on the project is Danica Like. She started in my lab as a high school intern and actually published the work she did as a high school intern in science. So she set the bar relatively high. Now, admittedly, we were one of 130 co-authors on that science paper, but nonetheless, Danica's first paper was in science. So we start working on this, and Danica starts to put together this flowchart. And the idea is we're going to follow through each of these steps, and at each point you can make a decision and choose your own adventure through the process.
Someone asked, what kind of decisions can you make?
Yeah, and so, for example, I just told you why we use sodium chloride. You can choose not to put sodium chloride. So is the DNA really charged enough that you don't get as good an extraction?
Or low sodium salt.
Or use sparkling water and find out why that's a bad idea for yourself.
Or use coffee.
Exactly. So the choices that we usually make are like, okay, we're going to do bananas or we're going to do strawberries. But each step, like, well, what happens if you don't mash as much, or if you mash more? And we're going to talk about this when we get a little bit further. The DNA is incredibly long. You can actually shear it and start to break those strands. And so there's a step later on where we start to swirl things. And oftentimes we get these like high school kids, and it sort of falls down on, so the kids are like, they do this quick buzz, and they get a more poor extraction because they've sheared the DNA.
Term coined in our paper, the Slobodian effect.
Because?
The failure to properly extract DNA.
One of the grad students in the lab, Mitchell Slobodian, for whatever reason, Mitchell can't do these extractions. And so, it was like repeatedly, it's like, hey Mitchell, will you help with this? Sure. And he would get the worst extraction out of the group. So we managed to get in the paper this individual variation in extraction. It's called the Slobodian effect. So the idea is at each step, you make a choice: salt, no salt, more mash, less mash. But so Danica is from Northern Ontario, and in the home she speaks French. She's francophone. She actually joined my lab. I was going to say she was at a French high school, but she was not. But when she joined the lab, almost all of her communication was en français, and I speak Duolingo French at best. But Danica came into my office one afternoon and said, we're putting together all of these resources. Would it be okay if I generated the resources in French as well as English? And it's a no-brainer. So we put it together in French, we put it together in English. Allie, why is that important?
accessibility to all students? Like, how many people on Earth speak English versus any other language?
Yeah. So, if you go into a classroom, and oftentimes teaching materials are in English, that's just sort of the nature of it, and most science is in English. We talk about representation, seeing yourself in whatever it is that you're looking at. Going into a classroom, to a French classroom with an English handout is not it's not as accessible, it's not as approachable, it's not as engaging. And so Danica's like, if I go back to talk to the kids that I speak French with, I want to be able to speak French and show this. So she started putting in the materials in French. I was volunteering with a group with GSA of young scientists, the Early Career Leadership Program, and so I went to one of our meetings and said, hey, we're going to do this DNA extraction protocol. The lead student who's driving this has said we should make it bilingual. Would anybody else like to participate? And so immediately we got Spanish, we got Arabic. One of the students in my lab at the time, Solomon, is from Ghana. And so Solomon translated it into Twi. So we've got the handout in Twi. We ended up with 10 languages. We also have it, so Jesse's got the Anishinaabe version. And so, sorry, you were going to say something?
No.
Okay, just, we've got 10 languages, and it's, if nothing else, like Laurentian's supposed to be a bilingual, tricultural institution. We very rarely hit that, where we have material that is available in the 3 cultures, 3 languages. So it was really important to put things in French, to be able to put things in Anishinaabe. Not every piece of the paper is in all the languages. All of the materials, the supplemental materials are available in French and English, and a lot of the material is available in the 10 other languages. So at the end of the day, we ended up with material that puts at least part of this exercise in the first language of 1.4 billion people, which is just, it's the way that we should be doing outreach. It's not always possible, but it's a really great start with something like this.
There's a question, what language is your dream language to add, and how many can you add?
So one of the things that I need to talk with the journal about is putting this in a way that's flexible. So language was our introduction into accessibility, but we published the materials in document format, not in PDFs, to try to allow people to take that PowerPoint, that Word document, and add onto that. And so you can download the materials, and then we encourage people to include any language. We were fairly far into the project, and I was talking to my department chair at the time, and he said, well, have you put it in Mandarin? I was like, I have not put it in Mandarin. And he said, the person whose office is right next to you speaks Mandarin. And so we got Guangdong and a grad student, sorry, to translate it to Mandarin. I think my dream language would be Gaelic. If we could get somebody to translate it into Cantonese and then somebody to translate it into Gaelic, that would just be pretty cool.
I think that we could get, so real languages, and that's the important bit, right? Like having people engage in their language with this material, that's the important bit. And we should get as many different languages as we can, like encourage people to translate it. I think that
say Klingon.
It'd be easy to convince some people who are into conlang, constructed languages like Klingon. There are different ones out there. There's like Elvish, like Tolkien Elvish. Like I think that
you're nothing but a hound dog.
Yeah, like Elvish, like exactly.
I take my answer back, and Klingon is now if we can get this translated into Klingon, we have not sufficiently hit the nerd world until we get somebody to
do DNA extraction in Klingon.
And Valencia smiled, so can you translate this for us into Klingon? I bet you can.
I have no knowledge of Klingon. But my wife, on the other hand.
Ooh. Okay, we should chat. So the handouts are a step in. They were sort of our gateway into accessibility. here's the handouts. It also allowed us to reach out to different labs. So we had, I think it's something like 10 different research groups that are part of the project. Most people worked in pairs, so that somebody would translate and somebody else would say, yes, that's correct. And some people actually, and I'm blanking, I think it was a student from, I don't see Sierra Leone, but one of the students said, we don't do enough science in our native language for me to be able to translate. Like this is what I speak at home, but I don't speak DNA extraction at home. And that was actually when we tried to put it into Anishinaabe. I had it the first time. What's the other half of Anishinaabe? Anishinaabe. The local Ojibwe language. A number of people said, this is just not the kind of vocabulary that we're used to, and we can't translate it.
Yeah, translating's hard because some phrases in languages don't translate very well, and some languages don't have the built-in vocabulary for exactly what a protein is. Like protein in English comes from protein, thing that changes, and that's why it got the name protein. So when you translate to another language, do you use that same logic of ever changing, and you try to have that quality? But where the word protein in English came from isn't necessarily the way that it's used now, so maybe it's not a good idea to use that in another language. You have to make choices when you're doing translations. And sometimes people are, it's difficult because you can translate one word multiple ways.
So there are a lot of issues with it. I mean, a simple issue is that a lot of people don't do science in their primary language. And so they're translating okay, well, I wouldn't translate it. I would do this in English. Okay, well, great, we've got handouts in English too. Okay, so we have been letting our DNA filter through. I think we're sort of at a point where it's stopped dripping, and let's do the next step. So we're going to carefully remove our filters. And at this stage, if something screws up, you just filter again. And when we're doing this with kids groups, if we've got 10 kids, 2 kids will have an issue and we just refilter it. So
I'm gonna try to get more.
And what are you doing?
So I'm just taking the filter and then squeezing the liquid out.
Okay, and so we're just gonna gently do this, but you can imagine saying to a group of 5- and 6-year-olds, gently squeeze the coffee filter. So when I've done this at science centers, they're like, yeah, you do not tell kids to do the squeezing part. But if you squeeze, you can get a lot of water out. All right. And so we've got this. Oh, does this work? What happens if I do this? Am I? Yeah? Okay. Yeah. So we've now got this solution, and you can see it starts to turn brown. It's just banana. And so it's putting the banana on the counter, and the banana starts to turn brown. And so it's just starting to oxidize a bit while it's...
Question from Chad is, where is the DNA now? Is it in the banana residue or is it in the water?
Right, and so the answer is at this point it's in both. So there's a lot of DNA in the material that we've left behind. The DNA that we're going to get is in the water that's here. And so we've left the solids behind, we're putting them in a cup. And so what we have is this banana solution. We've filtered out the big bits. And in the water now are banana cells. We've broken up some of the cell walls, the cell membrane is still intact. So you can think of this as being this sort of set of spheres, and inside the sphere is still those long threads of DNA. You got plenty.
I know, but I wanted to be really good.
All right. You do you. All right, so we've got banana cells now in the water, and we've got salt in the water that's ultimately going to coat the DNA. In cells, DNA is found in chromatin. We don't know what word source of chromatin comes from. Chromo, it's staining. So in early cell biology, there were these threads that were darkly staining that they called chromosomes. Chromosomes are DNA, but in equal amounts of protein. So it's about 50-50 DNA and protein. So when we do this extraction, we want the DNA, not the protein. So when we do this in the lab, we add commercial proteinase. When we do this on tabletop, we're going to add contact lens solution. So last.
So immediate question pops in. If I have contact lens solution, is it bad to use as a way to rehydrate my eyes if it's popping protein?
I don't know that. I am going to defer to the medical industry that has said that contact lens solution is okay. There's not a whole lot of proteinase in it, but one of the things that contact lens solution does is it helps keep protein films from forming on contacts, and so it has a bit of proteinase. We're going to just put a few drops in.
Yeah, I don't think you're actually supposed to like put this in your eye. It's just supposed to be like it goes in your eye, but like.
Yeah, so maybe it's not an eye wash.
No,
It's not like eye drops.
No.
And so at this stage, we're going to do the terribly clinically measure of somewhere between 6 and 10 drops of
1, 2, 3.
5, 6, 7.
Okay,
Okay, so, and this is another step. So we talked about what steps can you leave out. Oftentimes we don't do the proteinase, but it's a neat step that we can add in, and it allows us to talk about the fact that chromatin is made up of DNA and proteins. And so we're now going to gently swirl this. And now we're starting to break up those cells. So we want to be a little bit more gentle. We're no longer mashing because we don't want to start to shear those long strands of DNA. And so we've added salt, we've added proteinase, we've got water, we've got banana. Now we're going to go ahead and lyse those cells. And so we're going to add dish detergent. Any dish detergent you can use, absolutely any. I like the stuff that's colored because you can actually see it spread through the solution. So we'll just put in a couple of drops. And again, this is not rocket science. We just add a couple of drops and we get something that looks like this, where now we don't have a down shot, but if you look into it now, you can start to see these little drops of dish detergent. Okay, at this stage, things become fragile. And we're going to start to burst these cells, and as you burst the cells, meters of DNA are now spreading out from the cell. So in the cell, you've got the cell wall, you've got a cell membrane. Within that, you've got a nucleus. The DNA is inside the nucleus. Those different steps, the compartments are made up of membranes, and the membranes are made up largely of fats, lipids, and these detergents are going to break apart those lipids, separate the membranes, and lyse the cells, open up the cells. So we're going to swirl it just to mix that soap in and start to lyse the cells. Okay. And this is where we think the Slobodian effect often comes in, that you get kids that are really vigorously mixing through. And in the end, you'll get DNA, but you're going to get small pieces of DNA.
That won't look as fancy when you use that stick to pull up.
To pull up the DNA. Okay, so we're showing you how to do this. We're walking through each of the individual steps. We've got handouts that are part of the paper. At some point, Danica suggested, she came into my office again and said, hey, sometimes there are learners that really, they do better if they can actually see this instead of just reading about it. Can I make a video? Sure. Is it okay if I make 2 and we have one in English and one in French? Absolutely. So one of the things that Danica does outside of the lab is she works as a science communicator at Science North, the second largest science center in Canada. And she does demonstrations, and she does demonstrations in English, and she does demonstrations in French. So in the paper, there's a link to the YouTube videos that I think are on my lab page, maybe. And Danica goes through and does this entire extraction in either English or French, and walks you through the steps. As a teacher, a lot of the stuff you know, some of the stuff that you don't know, but having these resources, you can go and watch somebody do this and listen to Danica explain, what we're doing at each of those steps can be super important. So part of making this protocol accessible was making materials in different kinds of formats. Written format, handouts that people can print out, videos that you can go through and watch. All right, what's our next step? I think, okay, so we're now going to, we've mixed the samples, we've lysed the DNA. We now have in this solution long strands of DNA coated in sodium ions, coated in salt. And what we want to do is get them to clump. So we're going to mix gently the water solution, the aqueous solution here, with an alcohol solution that we're going to layer across the top of this. And what's going to happen at the interface of those two liquids is the DNA is going to precipitate. Alcohol, and you can do any alcohol. So we're using isopropanol. It's 99% isopropanol. It's just rubbing alcohol. And it's high percentage rubbing alcohol. If you do this at home, you need to be conscious of getting the high percentage isopropanol. You can get like a 30% isopropanol solution. Don't do that. So that you want to have a high enough concentration. In our local pharmacies, they're right next to each other. And so there's the low concentration, high concentration, just grab the right one. You can also do this with overproofed liquor store booze. And so depending on what state or country you live in, when I was a student in North Carolina, it was called Everclear, and it was overproof, essentially moonshine. In Ontario, we can get overproofed rum or overproofed vodka. It's about 70% alcohol instead of 35 or 40% alcohol. And this is what we do in the bars. So if we're doing this in a bar, we're actually we bring in our own alcohol because most bars don't keep overproofed booze. But it needs to be an alcohol, it needs to be relatively high concentration. If you do this with overproofed alcohol that can be consumed, I would strongly dissuade you from taking shots at the end of it. The protocol is actually designed so that when you do these saliva extractions, you can down it as a shot. Like there's nothing in there. A little bit of dish soap, some contact lens solution. It's all consumable. But overproof booze is 75%.
So it
ethanol.
cleans the spit you spit.
Yeah, and it's also a very harsh shot. Okay, so the difference between the two liquids is important. So we've got an alcohol and we've got a water. It's not exactly the polarity of the molecule, but it's very similar. And so it's called the isoelectric constant between water and alcohol is slightly different. And that difference means that there's a phase difference between the water and the alcohol. And the DNA, which is really comfortable being dissolved in water, will come out of solution at this interface between these two different solutions. And so that's why the difference is important. It doesn't have to be a particular kind of alcohol, but it needs to be an alcohol so that we've got this difference in dielectric constant between the two solutions. So Jesse, do you want to start at your end?
Am I remembering this correctly? But it also works a little better if this is cooled.
So
So if you're doing this at home, you can throw this or your overproof alcohol in the freezer for a little bit, let it cool down, and you might have a stronger effect.
That's right.
Oh, I thought that it mattered with ethanol versus isopropyl.
So if you're doing this with overproofed ethanol, so commercial alcohol, consumer grade, human grade alcohol, you do really want to cool it. And so I take the vodka bottles, put them in the freezer before we bring them in the classroom. With isopropanol, it seems to be more forgiving about room temperature. One of the punchlines with these steps is the protocol itself is incredibly forgiving. And so we have all these steps. You can use a different alcohol, you can use different temperature, and at the end of the day, you almost always get extraction. And so it's unless you're Mitchell Slobodian. Absolutely.
So I was being really careful not to pour this directly into the liquid. Instead, I was pouring it down the side so that it comes down and forms a layer. You know when you make those like fancy shots where there's like a little blue layer at the bottom and a clear layer on the top? I want to try to do something like that.
You you also really don't want to do this with a cup of coffee in front of you, because I just now reached for my coffee and inadvertently reached for the DNA extraction. So I am really, really, really horrible at lab safety, and yeah, so if you're doing this at home, put your coffee off to the side. You don't want to grab the wrong one. So I'm going to pour these. If you want to try to walk around and put that up to the camera, because in both of these, you can actually really see the DNA has already sent it out.
Folks are wondering how much alcohol did y'all need to add?
Yeah.
Well, you just need technically a layer above it, but
Can you see that? I don't know how.
Nice, yeah.
all right. So you just need a layer above it.
But so we
I have
often
more than
sort of
you.
approximate it by saying equal volumes. And so if you've got about, I'm still thinking in Canadian, so if you've got about 30 mils, so a few tablespoons of water, then you want to have about 30 mils of alcohol. As Jesse says, you just want the interface across that. And I will tell you, so if we just look at these, okay. And yet again, Allie wins. All right, so the last step that we're going to do is we're going to take anything. We use stir sticks. When we wrote the protocol, one of the things I wanted to be clear about is you're grabbing these incredibly long strands of DNA that are clumped together. If you use a plastic swizzle stick, it tends to slide off the swizzle stick. So these dollar store wooden stir sticks work really well because they've got just enough friction to sort of hold them in place. And so you want to gently swirl through, and this is, excuse me, often where we get the difference is you'll see a lot of, oh boy. Yeah, okay. So I've got a lot of DNA, but I'm having a hard time getting it actually to clump on my stir stick so I can see the DNA.
What about you, Jesse?
I can see it. I can see the thing I'm trying to grab. But I can't.
Yeah, but so if we do this, that's a resolution on that. You can see Allie's just got more. Now, it could be because I stirred too vigorously. If we do that, how am I doing? Okay. Or it could be because I've got a lower concentration. I've got more banana per unit of water. But okay, just like pause for a second as Jesse fights through his. This is DNA. Yeah. At some level, this is pretty amazing that this actually works. And the DNA that we've extracted here is from a banana, but as a molecule, this long chain polymer, it's exactly the same molecule in us as it is in a banana. So we do this with saliva. You're actually extracting your own DNA. When we do this bananas, we're doing the banana DNA, but it's exactly the same molecule. And this idea of life on the planet having a really complicated but essentially single origin, we all share the same genetic material. And so we're holding at the end of the stick this chain of information that's the same kind of chain of information that makes us us. So what makes a banana makes humans humans. You got a lot. Absolutely true. So what you find, so we do this in the classroom, you've got 20 kids, and so say to them, okay, who wants to do what? And so, half the kids can do isopropanol, half the kids can do ethanol. You can do cold ethanol, you can do room temperature ethanol, and you get different amounts of extraction. Actually, in the paper, Danica went through and did some comparisons where we actually quantified the amount of DNA that's there. One of the things that we're aware of is this is mostly DNA, but there's a lot of protein. That's why we add that proteinase. So we talk in the paper about ways that you can talk with the students about how would you purify the protein out of this. We did a comparison that I think is in the paper where we did a commercial kit that literally is made to extract DNA, and we compared that with these extractions. And not surprisingly, the commercial kit does a much better job, but these literally cost us pennies.
There's 3 questions that have come in. First, why did you use bananas?
Yep. So we use bananas because they're cheap, they're easy to get, and they're really soft. So once you peel them, that inner layer has a lot of tissue. I mean, the fruit has a lot of tissue, a lot of DNA. One of the things that we did in the comparisons, we can't do it in bananas, but we do this with blueberries. And we've got local blueberries that are just from the bush. So one of the things we should talk about is the importance of talking to different groups. I mentioned that Laurentian is tricultural, there's French, there's English, and then importantly, there's the local indigenous community. So we've got an indigenous sharing and learning center on campus. Allie and Jesse have both been part of events there. Allie's only in the lab because she saw Jesse speak at an event at the Indigenous Sharing Center. So when I've done some workshops there, we've used local fruit, and so we used local blueberries. We did a comparison where we took commercial blueberries and local blueberries, and you get a lot more DNA per mass, volume, out of the commercial blueberries because they're probably a really funky ploidy where you've got multiple copies of the genome in the blueberry. One reason that banana works really well is they're triploid. We're diploid, so we've got 2 copies of every chromosome. Bananas have 3. And they have a really, relatively speaking, big genome. And so any fruit will work, but some fruits work better than others. And these domesticated commercial fruits tend to have a lot of DNA and tend to work really well. It was interesting in that blueberry comparison because you could see the wild blueberry versus the domestic blueberry, that difference really showed up. But if you want to talk to a local community and you say to them, we walked outside and we collected these blueberries and we're going to extract the DNA from those blueberries, that's a different experience than here's a banana we got at the supermarket. Okay. So this is it. We've got it. We've got the DNA extraction. At this point, depending on what you're doing, we move on to different things in the classroom. Blah, blah, blah. But let's talk a little bit about, oh, wow, there you go. Okay, it worked.
He is just...
I'm not very good at scooping.
So there's a question about what you just scooped. How do we know that that is DNA and not goo?
So it is goo that is mostly DNA. And one of the ways we actually know what it is we know from other studies the wavelength of light that DNA absorbs, and we know the wavelength of light that proteins absorb. And so we know biochemically from other studies that this goo is made up of proteins and DNA. And so one of the things that Danica did for the paper that we do in the lab all the time is she took this DNA and she redissolved it in water and took samples of that and put it in a spectrophotometer, so an instrument that passes a known wavelength of light through the solution, and said how much of different wavelengths are absorbed. And so she did what was called 260-280. So she compared the amount of absorbance in the wavelength for protein with the amount of absorbance at the wavelength for DNA. And what she showed is there is protein there, like the commercial kits do a much better job pulling out the protein, but that a lot of what we're looking at is in fact DNA. And so we went back and analyzed the purity using this ratio of light absorbance between proteins and
so when you can take your home spectrophotometer and do that, and to confirm for yourself that you have only DNA.
We're not arguing that every step can be done at home. Sorry. So, but that's actually one of the ways the experiment is flexible. Is if you are doing this in a teaching lab, you'll have access to a spectrophotometer. If you're doing this at home with your kids, you'll say, okay, look, we know from other people's biochemical studies that what we're looking at right here is in fact DNA. Okay, there were some other things that we did to make the materials accessible. So do you remember the story of the handouts, the DNA handouts?
No.
Okay, so we started this journey with, we want to make a protocol so that anybody could do a DNA extraction either at home or in a classroom. Great. Danica had the really wonderful idea that it would be more engaging, accessible, approachable if we did this in multiple languages. Fine. And she came back and that we do it in different formats. So we have the video and we've got the the written stuff as well. So she comes back into my office another time and said, hey, I've been thinking about this, and there are lots of pictures of DNA on the web, but why don't we include a picture of the DNA that we say is pretty representational of the model that we have and the way that we think DNA works. Great, let's do that. And so she went back and was working on it. She came back and said, these models are often done in shades of red and green, and we know that red-green color blindness is relatively common, and so these diagrams are not as accessible if you have color blindness. So she spent a couple of days, maybe longer, and went through and found some web resources that allow you to optimize the coloring and shading of your diagram for the greatest number of people and color blindness. And so it's not 100% effective, but the diagram that's in the paper, so you look at the paper, there are a bunch of supplemental resources. If you pull up this diagram for DNA, it's in a very different color tone than most DNA diagrams, but it's because she has designed it to be accessible to people with color blindness. And I don't know what the numbers are. It's something like 10% of the population is colorblind. It's sex-linked, so many more men are colorblind than women. And when Danica came in and was telling me this, it was yet another embarrassing moment of why haven't we thought about this before? Almost a third of the people that I worked with going through my science education were colorblind. So one of my research undergrad advisors, I knew he was colorblind because he'd come in every day with the USA Today, which was the first color newspaper, and they always had these infographics. So Burgess would come in with the newspaper, and it had marker, or Sharpie on it, and it's because his wife would get the paper first, and she would code all of the infographics into black and white so that Burgess could actually read the paper. I was like, A, that's adorable, and B, that's really awesome, and something to think about. Another guy that I did my PhD with is also colorblind. So it wasn't coincidental that Joe wore shades of gray and khaki 100% of the time, in part because he once went out on a date wearing lavender corduroys because he couldn't see they were lavender, and 30 years later when I was working in his lab, was still traumatized by the lavender corduroy incident that his wife would bring up on a regular basis. But Danica went through and made the diagrams in colors and shades and shading that are accessible to a wide number of people, right? So it's this idea, once you start thinking about accessibility, and I'm sure there are things that we could do better. It's like, okay, we put it in multiple languages, we put it in multiple formats. What are things that we can do? And that's just something we have to keep thinking about as we go forward.
Mm-hmm.
So that's the extraction. That's what we've got there. You two came into the lab for a reason. Like there was something that brought you in. We do outreach. So we started this conversation with why do we do outreach? I feel like I've got a moral responsibility to tell the people that whose money I'm spending to do research what we're doing. We're not working on banana DNA, but this is a really great way to say to people, this is DNA, this is part of the step. We extract DNA in the lab on a regular basis. It's a way to have that conversation. How did outreach impact the two of you with deciding to go into your graduate students, your professional scientists? What does outreach mean to you?
I mean, I think that I would like to inspire more people to go into science, science specifically, but like any of the STEM fields, technologist, engineer, mathematician. I mean, sometimes in math, in mathematics, they find something in there that is applicable to your own research. So having mathematicians is really important. But I would like to inspire people to go into science. What about you?
Representation matters, and that's something I greatly lacked growing up. So I think that my interest in science and pursuing research as a career, I didn't see that ever, especially for Indigenous women. So it's really important to me to try and be that representation and give back to my community as much as possible.
Yeah, and Allie had a grant last year from the federal government to go back to your community. So what does that mean to you?
that to me. So I recognized a lot of the kids that I was giving these talks to, and a lot of them knew my sister. So like they knew who I was, but not necessarily know what I'm doing.
Yeah.
So it was really important for me to not only say like, this is what research is, because that's something that isn't inherent. We didn't grow up, or we didn't learn, like that's not what you learn. And different careers, research was never on my radar at least. So just putting it out there is really impactful, I think.
Yeah.
So we're here at the Allied Genetics Conference. And yesterday we went and saw a series of talks on science outreach. And one of the takeaways of one of the presentations was that they work with like fourth graders with one of their outreach schemas. And their reason for that is that they found that people who go into science usually have a formative experience with science at about fourth grade, at about when you're about that age. And Allie, do you think that you had a formative experience with science that encouraged you to go into science?
I think my formative experience happened a little later. So in about 8th grade, I remember doing a hands-on activity and like dissecting. And I think that is what inevitably led me to bugs.
Thomas?
Yeah. So, I mean, I started this by saying I've known I was going to be a professor since I was 6, right? But so one of the things that I do actually alongside this DNA extraction is I have participants in workshops or any of my classes, I have them draw a scientist. And there's a draw a scientist project, it's a big international project, incredibly well done. And what people have found over and over again is that people tend to draw, so you ask them draw what you think of when you hear scientists, and they draw stereotypes, they don't draw themselves. So when I do this in a classroom, most of my classrooms are equally split between men and women, and I will get maybe 10% women in the drawings.
It's been going up.
Yeah, I hope it's been going up. I'm not sure, but it's always not. It's never been 50%. So I've been able to see myself as a scientist since I was a little tiny kid. Like I had children's books that were, essentially Tommy the Scientist kind of thing, and I could see myself there. We've got a lot of middle-aged white guys in science. That's an easy demographic to see. But it's important that there are other faces that we get out there.
Yeah.
Yeah, I didn't see myself as a scientist until I was told that I was one. And when I did that activity with you, I too drew a male. I never saw myself as a scientist.
Yeah. So these kind of outreach things are really important for seeing yourself. So we've talked about this before, how few Indigenous faces you saw at the front of the classroom as opposed to in the back of the classroom. I don't remember, where did you go to school, Jesse?
I went to school just in Sudbury.
Okay.
So went to school in Lively, St. James, went to a Catholic school. Then in high school, another Catholic school, St. Benedict's. Then I went to Laurentian.
But you grew up on Rose?
Yeah, but so the reserve I grew up on is essentially just a suburb in the town.
Okay.
In Sudbury. It's not exact. It's not what most people would think of as a reserve.
Okay. You grew up in a community, so not on reserve.
Yeah.
But do you know what the percentage of Indigenous population is?
It's definitely changed, but growing up, it was probably above 90%.
So 90% Indigenous, and you had 2 teachers who were Indigenous.
Yep.
The entire time you were in school. So we're not getting that reflection of the population in the front of the classroom.
Oh. So representation in media is important, right? Like, being able to see yourself in these roles, like be able to identify, like, oh, I can imagine that's me. So my formative experience with a scientist was from a game that me and my brother was playing. I was watching him play because I was a little kid. And it was more just the representation of like scientists as a job, as like a thing, what inspired me to become a scientist. So I think positive representation of these fields in media, I think that's very important.
Yeah,
That's a great point.
and it's, so I've known I've been to be a scientist for a long time, but one of the turning points was watching the Indiana Jones movies and like, hey, scientists can not only be nerdy, which I had that down pat, but they could be cool and nerdy and colonial and many, many not so awesome things in retrospect. But like the number of scientists, if you talk to me, like, yeah, Indiana Jones is why I went into science. Like, oh God, yeah, I did it. Like, hands up if it was Indiana Jones. Yes, it was Indiana Jones.
It's akin to, I drew Albert Einstein.
There you go. Yeah, so when we do the draw scientists, we tend to get more Albert Einsteins in the population of drawings than we have women. And so I put 3 categories on the board: male, female. We now put a 4th because a lot of kids have been drawing non-gendered scientists, which is a pretty awesome change that's come up in the last few years. And then the last category is Albert Einsteins. And there are usually more Albert Einsteins than there are female scientists. So yeah, there are lots of aspects of outreach that come up in this publication and are reasons why we tried to make it as accessible as we did. Representation matters. Seeing your own language in the DNA handouts is a big deal, whether it's Urdu or Arabic or Anishinaabe or English or French. And hopefully at some point Klingon.
Question from the chat is, thank you so much for this conversation. What advice do you have to me, who also am not part of the classic scientist category, and how can I get past this feeling of not belonging and not being able to pursue that education?
So I felt that a lot. And I think my confidence has grown just doing the science, but there's still in the back of my mind, I'm not qualified enough, I'm not good enough, I'm not what have you, right? And at the end of the day, none of that really matters if you're having fun. And that's kind of what keeps me going, is I may feel these doubts about myself, but I really, really enjoy what I do. And that to me has gotten me through my master's and soon to be into a PhD.
Yeah, just to be clear, you're not through your master's.
Well,
just to be clear,
through
you got to
the
defend.
first year and a half of my master's, and then soon to be through my master's.
And it's going well.
Yes.
I will tell you that I do not know that I've ever worked with a student that is more excited about their results than Allie. And as a major professor or primary investigator, organizing a research group, 99% of what I get is this didn't work, or I need you to buy this. And it's like a constant push to say to students, text me the good things too, so that if your name pops up on my phone, it's not like, oh, I wonder what broke this time. And Allie has texted me at 10 o'clock on a Friday with a screenshot on the phone. Look at this graph, look at the tendency here. And the one that was still my favorite is, hey, I was just sitting around and it occurred to me, so I graphed all the data from when I was a 3rd-year undergrad through the first year of my master's, and look at the trend that's across this data at 10 o'clock on a Friday night.
Yeah.
I'm like, yes, you are a nerd.
Yeah.
And it was a super cool graph. It was super cool. So yeah, I mean, lots of people struggle, and even people that you don't think should, with this feeling of not being part of science. These kind of experiments, I think, can get your hands dirty, let you feel part of that. I think globally science is doing more citizen science. And Jesse, you were talking about this project. We're actually going to start collaborating with them. And was that a different project?
It was the one started in Spain.
So I'm confounding kind of projects. But the idea is to bring people in at a really young age and get them doing science so that they can see science, either see themselves in science, or just demystify the idea of science in general. and that, in the climate that we live in at the moment, demystifying science is as important as allowing people to see themselves in science.
Yeah. And actually, I think that's another thing that I would like to accomplish with outreach. As much as I want to encourage as many people as I can to go into science, not everyone has to be a scientist. We need society to keep going. If everyone was a scientist, I don't think we'd get any plumbing done. But I think that it's important that everyone understands science. And so in my community, we're wanting to have our own research group. So rather than having someone outside of our communities come in and research us or research our things on happening in our lands, instead we would like to be doing that research for ourselves. But part of that comes with having to convince my communities that this is a good thing to do and that we should be doing this, and part of that is I think that I need to inform people about science, and that you can do a lot of bad things with science. You can do a lot of good things, but science itself isn't good or bad, right? It's how you use it or the way that you do it that can be harmful or helpful. And I think that I need to communicate that science is a useful thing to use in trying to understand the world to my communities. And part of that is you just need to engage people in science and let them understand it.
Yeah, I think one of the lessons for me as we're going through this project and other things in the lab and interacting with different students in the lab is appreciating, depending on where you're from, science may have a better or worse reputation. And if you're a middle class kid from Connecticut, science is great. If you're an underrepresented kid from Northern Ontario or from anywhere, especially in populations that have not benefited as much from science,
Or have been harmed by science.
this is a really big deal. And so going into communities and talking, and for me to go into a community and talk is one thing. For people that are part of the community to go in and have conversations. So Allie had a grant from the federal government. Last year, Jesse is working on a grant for this coming year. But one of the things that came up when we were discussing your project is how do we make that project long-term and not just a short-term, here's a Band-Aid, here's a fix.
Yeah.
This is a long-term thing that we're going to invest in relationships. So do you remember I don't think we've come up with a solution with that.
Not yet. So I've been mulling it over. I think that the last time we had a conversation with North Shore Tribal Council, we were coming down on maybe doing a once a year thing, maybe like one day or a few days during the summer. So SHAD is a week-long thing, but I think a smaller, more concentrated version of that or something like that could be useful. The other thing is we could do a annual trip down North Shore, do a small science camp. And actually what I was thinking is that with this new project that we're hopefully going to become part of, originating in Spain, it's a Drosophila, but now hopefully Canada and the United States, where they're collecting local fruit fly populations, Drosophila populations, and they're trying to see what the landscape of Drosophila is in the world. And part of that is you have to do science and you can engage people in doing science. So I wish I had a good grasp on this. I'm going to explain this to you, but I don't know if I have the tightest grasp on it. But in essence, you're going to be getting youths collecting Drosophila from the environment and then taking those and sending them off or sending them to a associated research campus to do sequencing and analysis of those Drosophilids.
But yeah, the punchline of that is that it's an ongoing project that's going to be done year after year after year that engages kids really young, and then also re-engages them in high school. So there are multiple steps. So we may end up playing it as part of that as well.
So then my idea was that make it a, annual summer camp that involves this project.
Yeah. So I want to wrap up with the two of you because I want you to go hear the talk on Copper Toxicity that's coming up. When we put this project together, we had a lot of people come together as part of the lab and do this. Did the inclusion thing mean anything to you? Did it mean anything more or less beforehand, afterwards?
The inclusion.
Well, just the idea of making this project more inclusive. So Danica was really pushing for this initially, but what does inclusion mean to you?
I think it
Well-
goes to representation, and not only just like physically, but in the literature, right? So for the paper, if you can look at a resource and be like, oh, this is in my language, it's much better than just being like, okay, well, we think about science typically in English.
Yeah.
We're going to continue like that. So,
Well, for me, it more makes me lament not knowing my own language.
yeah. Yay, colonialism.
I mean, Canada did a bang up job of
Yeah.
destroying my culture. It's pretty great.
Yeah.
Okay, yeah, sorry. As a physical representation of everything that's been wrong in North America for the last 400 years, I apologize. Yeah, I mean, that's not a trivial point. It is, there are upsides and downsides to this, and one of the downsides was realizing how little science is actually accessible outside of English.
Yeah.
And that's a really big deal. I want to thank the two of you for doing this, for giving us your time and your perspective, not pulling a Mitchell, and the Slobodian effect did not rear its head in this one. If you two would make it to that talk, that'd be super cool, so that we can find out more about copper toxicity.
Science is really cool.
Yeah.
I'm going to stay, and we've got other questions we can
We can wrap up now, and we've
Okay.
kind of cleared out all the questions. Y'all have been actually hitting the questions that have been coming in as well. So
All right, super cool. All right, thanks everybody.
Thank you.
Thank everyone.